When the human body survives a threat, it shifts into a period of systemic physiological recovery. Heart rate drops, digestion activates, pupils constrict. This coordinated cooldown is executed by the parasympathetic nervous system. Its mandate is highly specific: force the body to rest, digest, constrict, and empty. Executing that mandate requires a biological contradiction. The system has to command cardiac muscle tissue to relax while simultaneously forcing bladder and intestinal muscle tissue to contract. To manage these opposing commands across the entire human anatomy, the body relies on a highly conserved circuit. It uses exactly 2 neurons in sequence, communicating via a single neurotransmitter. This chart illustrates the complete parasympathetic architecture. It is a precise craniosacral map. The physical wiring originates in the brainstem and the lower spine, and the different physiological responses are determined by the receptor subtypes at the terminal organ. By controlling dozens of physiological variables with only a handful of chemical components, the parasympathetic network operates with high biological efficiency. Down at the cellular level, the system relies on a specific molecular messenger synthesized directly within the nerve terminal. This molecule is acetylcholine, or ACH. It is manufactured when a precursor molecule, choline, fuses with acetyl coenzyme A. This synthesis is catalyzed by the enzyme choline acetyltransferase, and the resulting ACH is packed into vesicles, waiting for a signal. Release requires voltage. When an action potential reaches the terminal, calcium floods inside. This calcium influx forces the vesicles to fuse with the cell membrane, releasing acetylcholine into the synaptic cleft. This chemical release drives the universal parasympathetic flow. Every signal begins with a preganglionic neuron exiting the central nervous system. On this isolated column of our map, we see the first relay. The preganglionic neuron projects outward and releases acetylcholine onto an autonomic ganglion. At this ganglion, the receiving hardware is universally a nicotinic receptor, specifically the NN subtype. When ACH binds here, it triggers rapid depolarization, continuing the electrical signal. The signal travels down the short postganglionic neuron, which reaches the actual target organ. Here, it releases acetylcholine a second time, but the terminal organ relies on a different piece of hardware, a muscarinic receptor. This repeating chemical relay, preganglionic nicotinic to postganglionic muscarinic, forms the foundation for every parasympathetic action in the body. If the chemical signal is always acetylcholine, we have to explain how the same neurotransmitter causes the gut to churn but the heart to slow down. The divergence happens within the cell membrane of the target organ through specific subtypes of G protein-coupled receptors. Track the M3 receptor. When ACH binds to M3, it couples to GQ proteins. This activates phospholipase C, generating IP3, which triggers the release of calcium from internal storage. A calcium influx floods the cell, physically driving smooth muscle contraction and glandular secretion. The M2 receptor executes a different logic. It couples to GI proteins, which inhibit the production of CAMP. This triggers potassium channels to open. Positive potassium ions rush out, hyperpolarizing the cell and stalling electrical activity. The incoming nerve signal does not dictate the physical response. The physiological outcome is hard-coded by the target organ's local receptor hardware. Applying these molecular rules to gross anatomy reveals a top-down routing network. We begin with the cranial nerves exiting the brainstem. Tracing this blue line on the anatomical chart, cranial nerve 3, the oculomotor nerve, originates at the Edinger-Westphal nucleus and projects to the ciliary ganglion. Once the postganglionic neuron reaches the eye, it hits M3 receptors. The resulting calcium influx contracts the sphincter pupillae to constrict the pupil and contracts the ciliary muscle, thickening the lens to allow for near vision. Moving down the brainstem, cranial nerve 7, the facial nerve, exits the superior salivatory nucleus and splits toward the pterygocalytine and submandibular ganglia. Following these green paths on the chart, the facial nerve targets M3 receptors to trigger fluid production, tears from the lacrimal gland, plus watery saliva and nasal mucus. Cranial nerve 9, the glossopharyngeal nerve, fires from the inferior salivatory nucleus, using the otic ganglion as its relay station to reach the parotid gland. The parotid gland is lined with M3 receptors. Activation here results in major salivation, prepping the digestive tract. The architecture of the upper cranial nerves relies exclusively on the M3 pathway. They force secretory and constrictive preparations across the head and neck. The thick orange tracts dominating this chart represent cranial nerve X, the vagus nerve. Originating from the dorsal motor nucleus and nucleus ambiguus, it is the primary parasympathetic conduit for the internal organs. Notice the target receptors on the heart. Vagal branches reach ganglia located on or near the SA and AV nodes. Here, the postganglionic neurons deploy acetylcholine onto M2 receptors. Because M2 receptors couple to GI proteins, they cause hyperpolarization. This slows phase 4 depolarization in the cardiac tissue, dropping the heart rate and inducing bradycardia. If a patient requires an artificial increase in heart rate, administering the drug atropine physically blocks these M2 receptors, severing the vagus nerve's braking mechanism. Looking at the lungs on our schematic, the local hardware switches. The vagal branches hit M3 receptors in the respiratory tissue. The M3 cascade forces bronchial smooth muscle to constrict and goblet cells to secrete mucus. In respiratory distress, the drug ipratropium is used to block this specific M3 mechanism, keeping the airways open. The final major vagal branches deliver routing to the gut. Across the stomach, intestines, pancreas, and gallbladder, M3 receptors dominate the landscape. This triggers an upregulation of the gastrointestinal tract. Smooth muscle motility accelerates, acid and digestive enzymes spike, bile is released, and the internal sphincters relax. The vagus nerve operates as a master split cable. It downregulates cardiovascular output using M2 receptors, while simultaneously upregulating respiratory and digestive function through M3. The brainstem cannot wire the lower pelvis alone. As shown on the bottom left of our chart, a secondary origin point emerges at the terminal sacral cord, spanning segments S2, S3, and S4. Tracing these blue tracks shows the sacral nerves routing directly to their visceral targets, the distal colon, the bladder, and the reproductive organs. Urination relies on a precise M3 mechanism. The parasympathetic signal must force the outer detrusor muscle to contract, while simultaneously commanding the internal sphincter to relax. This same sacral outflow controls the smooth muscle contractions required for defecation and drives the vascular engorgement necessary to achieve an erection. The sacral nerves complete the anatomical map. They ensure the physical expulsion of waste mirrors the restorative intake driven by the cranial nerves above. The internal reach of the parasympathetic network clearly illustrates the vast scale of the body's craniosacral topology. This blueprint demonstrates how systemic recovery is managed from the poles of the central nervous system. It funnels acetylcholine through an unvarying 2-neuron relay. By splitting the terminal response between M2 and M3 receptors, the body balances competing physiological demands with precision. Systemic recovery is the functional output of this highly conserved biological routing.